All right, so we'll begin now. Hello everyone. I hope everyone is doing well today, and welcome to our third session for the summer crash course on meta-analysis. My name is Zahid Asghar, and I will be the instructor for today's session. So in our previous session, we discussed what the grow and the path explorer functions were in IPA. The grow function allows us to see all of the related molecules to the node that we're analyzing, and the path explorer function allows us to see what the intermediary nodes are between the two nodes slash molecules that we're analyzing. And in today's session, we're going to be going over how we can filter our searches, what the shortest path and what the shortest path plus one is, the different layout options that IPA provides for us, such as circular, hierarchical. We'll be going over more of these later on in the session. And we will also be going over how we can export our data from our pathways. So before we begin, I just like to mention again that there will be a quiz at the end of the session. The quiz does have points that go towards, if you come in first, second, or third place, towards the final quiz that takes place on the 31st of August. And there is a monetary prize for the final quiz as well. So before we delve more into it, right, we'll just review the task from the last session. So if you remember from our last session, I did ask you to use the grow feature to grow familial tuberous sclerosis, which is a genetic condition that leads to skin and systemic findings, and specify that for dermatologic disease. So if you went ahead and did that, you should have gotten something that looks like this. So we have several molecules that go off of familial tuberous sclerosis. So we have three going downstream. So downstream meaning things that familial tuberous sclerosis has an effect on, these being mTOR, PKD1, and we have three that are upstream, arrows that are going towards familial tuberous sclerosis, meaning that they have an effect on familial tuberous sclerosis. These being TSC2, TSC1, these are just basically genes that predispose to this genetic condition, and PIK3CA. So these were what you should have found after using the grow feature and in IPA. And additionally, I also ask you to use the grow feature for methylmalonic acid as well. So upon using the grow feature for methylmalonic acid, you should have created something that looks something like this. Several arrows going up that are upstream, arrows that go up to methylmalonic acid. These affect methylmalonic acid. And several arrows going down. These are downstream to methylmalonic acid. So you should have gone something that looks like this. And again, these are after I've already moved around the nodes. Like I mentioned in the previous class as well, you're able to move around these individual nodes and create it into something that looks more visually appealing. In this session as well, I will also be talking about layout options that IPA provides for us. So you're able to move it around, but also IP provides for you different layout options. It really depends on what you are looking for, because sometimes the layout options that IPA provides for you may not be exactly what you're looking for, but there are layout options available for you that IPA provides, and we'll be going over them. And depending on your situation, what you're intending to do, right, you may choose to use the layout options that IPA provides. So we will be discussing that later on as well. Okay. Finally, if you use the Path Explorer for methylmalonic acid and homocysteine connected to vitamin B12, you should have gone something like this. It's quite a few connections over here, as you can see, and it connects to cyanocobalamin. Cyanocobalamin is another term for vitamin B12, basically. So you should have gone something that looks like this. And using the Path Explorer feature for familial tuberous sclerosis as a dermatologic disease, connected to angiogenesis. Angiogenesis is the creation of new blood vessels. So upon doing that, you should have gone something that looks like this. So familial tuberous sclerosis here, angiogenesis over here, the connections. And again, I've rearranged the nodes make it look a little bit more visually appealing. All right. So again, like I mentioned today, we will be going over the how to filter our searches, the different filter options that IP provides. And we can filter our searches both for the grow feature and the path explorer feature as well. So both of them are able to be filtered. The different layouts for our data. So that being the circular, hierarchical, there's several that we will be looking into in today's session. The shortest paths and the shortest paths plus one, we will be explaining that more in detail later. and how to export your data. All right. So before we delve into that, we'll just do a refresher, quick 5-10 minutes on how the grow feature and the path export feature work, in case you may have forgotten from the last session, or just as a general refresher as well. So basically the grow feature, it allows you to visualize all the related molecules to the node that you're analyzing. And that includes both upstream and downstream. So upstream being molecules or nodes that affect the node that you're analyzing. So in the case of, if you go back to the case of familial tuberous sclerosis, so TSC2, TSC1, these are genes that eventually cause familial tuberous sclerosis. So these are upstream of it. They have an effect on this node, okay? And downstream being these right here, like mTOR, PKD1, familial tuberous sclerosis has an effect on these downstream nodes. So, in growth function, it shows you upstream and downstream, right? And it shows you direct, indirect relations, chemical interactions, activation, and inhibition relations. If you remember from the last class, the different types of lines that connect different nodes, not all of them are congruous, right? And they basically indicate different types of relations or different types of interactions. So a solid line indicates a direct interaction. A dotted line indicates indirect. Chemical interactions are different. Activation is an arrow. Inhibition is a straight line with a little snub sort of at the end. So again, it shows you all of these different types of interactions as well, right? So the example that we used from last time was with buprenorphine. So, just as a refresher, we type in buprenorphine. Then we can either click enter on our computer, right? Or if it shows up over here, like buprenorphine shows up here, we can also just go ahead and click buprenorphine separately, right? But in this case, we'll just click enter, right? And upon clicking enter, it shows us all the related search threads to buprenorphine. So we have buprenorphine / naloxone, buprenorphine containing protein products, sorry. So we have all these related search threads to buprenorphine. We'll go ahead and select buprenorphine over here because that's the one that we want to add to our pathway. So we'll go ahead and select it over here. We'll add it to our pathway. We'll add it as a symbol because we do not want the related molecules to buprenorphine in our pathway. And we'll click OK. And it shows up on our pathway like this, right? Then we can go ahead and click on the build feature. So we click on build, we select a tool, we select for grow, and then we can select for if you want to include upstream or downstream molecules, how many we want to include in our pathway, right? And once we've selected for everything that we want, we click apply over here, and we get all of our related molecules over here like this. And we can then go ahead and rearrange them as well. So again, like we went over the general settings in our last class, we will also be going over these different settings as well that you're able to filter your data by, right? All right, so we can go ahead and like rearrange these nodes, make it more visually appealing, move them around, right? And then we also discussed the path explorer feature as well. And basically the path explorer feature allows us to connect two molecules / nodes to one another and determine any relations that they have with one another. So all the intermediary rather nodes that are in between those two nodes. So for example, like if you take let's say vitamin B12 to let's say some disease state. Okay, so say between vitamin B12 there's NF kappa beta, then after NF kappa beta there might be calcium, and then after calcium there might be let's say aplastic anemia. Okay, just as a rudimentary example. This isn't the case in real life, but just as an example. So like it'll show you all the intermediary things. Okay, after vitamin B12 comes NF kappa beta. After NF kappa beta comes calcium, then comes the disease state. So it shows you all of the intermediaries that eventually lead to what you're analyzing. And again, it shows you the direct, indirect relations, chemical interactions, activation, and inhibition relations, just like the growth function as well. Okay. So the example that we used last time as well was with buprenorphine. We search for buprenorphine here, right? We can either then click enter, or we can select buprenorphine if it shows up over here. So in this case, we'll just click enter on our computer. Upon clicking enter, it takes us to all the related search threads. Then we can, select for whichever we like. So in this case, we'll just select for buprenorphine here. We'll click on add to my pathway. We'll add it as a symbol over here, and we'll click OK. Then it gets added to our pathway. Then we'll also add apoptosis because we're going to be comparing it to apoptosis, whatever effects buprenorphine might have related to apoptosis. So let's type in apoptosis over here. Then we can either click enter, or we can, select it from over here if it shows up. So in this case, we'll just click enter, and that shows us all the related search threads to apoptosis. We'll select the top one over here. We'll add to our pathway. We'll add it as a disease or function, and then we'll click OK. And both of them are now added to our pathway. Then we can go onto the build feature. We'll select a tool. We'll select for Path Explorer. And that takes us to this page over here. So we can then go ahead and add buprenorphine to set A or set B. So basically, these are comparing, we can add either buprenorphine or apoptosis to either set A or set B. It does not quite really matter. We'll just add buprenorphine to set A over here, right? So we'll select for buprenorphine. Upon selecting, it turns blue like this. So that's how you know it's been selected for. We'll add. We'll do the same thing for apoptosis. We'll select it. It turns blue. We'll add it to our pathway. Then we'll set the direction. So the direction can either go from set A to set B, so showing you all of the interrelated nodes / molecules going from buprenorphine to apoptosis, going from set B to set A, which will show you all the interrelated nodes / molecules going from apoptosis to buprenorphine. Or we can go any direction, which will show us both. So showing pathways or relationships that go from buprenorphine to apoptosis and apoptosis to buprenorphine at the same time. So we can select for either one, but in this case, we'll just select for going from buprenorphine set A to apoptosis set B. Okay, so this one right here. We go ahead and apply those changes, and it shows us all of the related ways that they found, all the related relationships that IPA has found. We can either select for individual pathways, because again, oftentimes, once you use IPA a little bit more, you will find that oftentimes there will be hundreds, thousands of different, pathways that IPA finds for you, and including all of them may not be practical. So it allows you to select for which ones you'd like to include. So you're able to go ahead and look through all the different ones and which ones you'd like to include into your pathway. In this case, we'll just select for all of them. Okay, so we select this, that selects for all of them at once, and we'll add that to our pathway. And we get something like this. Then we can go ahead and rearrange the different molecules and make it look a little bit more visually appealing like this. And we get this, okay? So now we'll be going over how to filter our searches. So in our last session, we only really dealt with the general settings, but there are other filter options that are available. These being data sources, confidence level, species, tissue lines, mutations, relationship, etc. Right? There's multiple different filter options that are available. And why is it important to filter? So, one of the things that we discussed in our first session was that systematic reviews and meta-analysis, they often have inclusion and exclusion criteria. So you can't just include everything into your research. You have to specify for what specifically you're looking for, right? So it's necessary in that sense for systematic reviews and meta-analysis. Oftentimes you need the most recent literature, right? So you might want to specify for the publication date. Sometimes you're looking for a specific organelle. For example, specifically you're looking at macrophages as an example, right? So you want to specify for macrophages, right? The other thing is that, like I mentioned before, there's oftentimes many different connections that you create, right? So, that becomes very difficult to sort through. So by filtering, right, that narrows that sort of filter search, right? So that's another reason for why you might want to filter. And additionally, the results may differ before filtering versus after filtering. So as an example, with morphine connected to, let's say, interferon gamma, okay, which is an inflammatory cytokine, okay? Say prior to filtering, you might find that morphine has an inhibitory effect on interferon gamma. But after filtering, say you specify for macrophages, you specify for publication dates between 2000 to 2023. After filtering, you find that, okay, it actually has a activating effect on interferon gamma. So again, like this, right? Like once you specify results, you find that it has a different result. So that's another benefit to filtering. That's one of the reasons why you would end up filtering your search results. So we'll go over the different filter options that we have. So beginning with data sources. Actually, before that, like just an example of how filtering might look, right? So over here we have morphine. So this is morphine prior to being filtered. So we can see here there's a total of 307 relationships, right? Prior to filtering. Upon filtering, we find that upon filtering for macrophages and publications between 2000 to 2024, we find that there's a total of 23 relationships. So again, it's narrowed down drastically from 307 to 23. So again, like just as a periphery, right? Just keep in mind, it's not always the case that there is a reduction in the number of intermediary molecules, right? I have had in my previous research where upon filtering, there's in fact more, right? And that's just related to the algorithm of IPA. But generally speaking, upon filtering, there is a reduction in the number of molecules that you find related that IPA produces for you upon using the grow feature or the path export feature. Okay, so the first filter option is the data sources. And the two major data sources are either from IPA's data sources itself or third-party sources. So IPA has its own data sources that it has, and it also employs third-party sources as well. So you're able to choose from either or in your data. So the IPA's own data sources include the Ingenuity expert findings. So we see over here the Ingenuity expert information includes the Ingenuity expert findings, as well as the Ingenuity expert assist findings. So these are IPA's own data sources. So you can select from either expert findings, expert assist findings, or both at the same time, right? You have that option. And in addition, there's also third-party information, which is not directly from IPA. IPA has partnerships with other companies, with other data sources that they also pull their information from. So you're also able to either include that information or exclude that information. And the options that they give you for including or excluding are microRNA, mRNA interactions, protein-protein interactions, and as well as additional sources. These additional sources include things such as the mouse genome database and the drug bank, etc. This is not an exhaustive list by any measure, but these are some examples of additional sources that they include. Additionally, you can also what's called filter by the confidence level. So confidence level is basically a statistical term that basically is the likelihood that the statistical results you have actually reflect reality. So you can include results that are experimentally observed. So, results that are actually founded upon experimental evidence, right? Results that have a high predicted level of experimentally soundness, right? The likelihood that like the results will actually reflect experimental findings as well is high, or the likelihood that the findings that you're including will have experimental sounding is a moderate prediction. So you can include things that are experimentally founded, they're proven to be experimentally founded, have a high likelihood of being experimentally founded, or a moderate prediction of being experimentally founded. So you're able to select for that as well, right? You're also able to select for these species. So that being humans, mouse, rat, or uncategorized. So humans, like we discussed, this would be our clinical trials, right? And mouse, rats, this would be our in vivo studies on animals, right? So we're able to include that too. And also uncategorized. So, animal studies that may not be on mouse or rats, right? You're also able to select for those, right? So that's another filter category. You're also able to select for tissues and cell lines. So if you go on tissues and cell lines, you'll see that there's tissues and primary cells as well as cell lines, right? So the tissues and primary cells and cell lines, right? Primary cells, you will get something that looks like this. Once you open up the window, you'll see that there's tissues and primary cells not otherwise specified. There's cells, nervous system, organ systems, other tissues, and primary cells. So examples of cells that you might be able to select for would be things such as adipocytes, bone marrow cells, endothelial cells, etc. The list goes on. There's quite a few. You're also able to select for the nervous system, so things such as the brainstem, hippocampus, amygdala, cerebellum, etc. Right? And you're also able to select for organ systems right here. So things such as the heart, the kidney, the liver, etc. Right? And you're also able to select for different cell lines. So these primarily include like cancer cell lines, such as breast cancer cell lines, cervical cancer cell lines, CNS cell lines, colon cancer, etc. Right? But you're also able to select for different cell lines that have been cultured in laboratory settings as well, right? So you're able to either include those or exclude those as well in your filtering, right? And you're also able to select for mutations. So basically relationships that might be related to a certain type of mutation, such as a wild type mutation, translational impact, inheritance mode, right? So you're able to select for specific mutations that some relations might result in, right? So you're able to select for that. And you're also able to select for the different types of relations. So like we discussed, there's activation, there's inhibition, there's a correlation expression, these different types of relations, right? So you're able to select for these specific relationship types and either include them or exclude them in your data as well. Okay. And also publication date range. So, for the date range that they provide is from January 1927 to December 2024. So you're able to basically select what date range you want your publications to range from and to, right? So, and basically this is again important for our inclusion and exclusion criteria, often for systematic reviews or meta-analysis, because oftentimes they may require the most recent literature. So you're able to specify for that in your filter options, okay? And you're also able to specify for the types of nodes. So for example, like, we've looked at diseases, we've looked at functions, we've looked at chemicals, we've looked at drugs, right? So the different types of nodes that you want to have in your pathway, you're also able to select for those different nodes. So there's a long list. This is obviously not an exhaustive list. It does go down further, right? But you're able to select for the nodes that you're able to select for. So whichever nodes you want to have in your pathway, you are also able to select for that. And you're also able to select for diseases. So whichever molecules you want in your pathway that have a relation to a different type of disease. So for example, if the molecules you want in your pathway have some sort of relation to auditory disease or to cancer or to cardiovascular disease, right? So you're able to include the molecules or nodes that have a relationship to those specific types of disease, okay? And you're also able to select for biofluids. So these are basically whichever biofluid the molecules that will be included in your pathway can be found in. So say the molecules in your pathway can be found in the blood, they can be found in saliva, they can be found in sputum, they can be found in the cerebral spinal fluid, right, etc., right? Whichever biofluids your molecules are found in, right, you can specify for that as well. So only molecules that are found in blood, or excluding molecules that are found in blood, right? You're able to do that. So we'll just look at an example of how we would go about filtering. So in this example, we have morphine and ketamine, and we are connecting this to NMDA. So NMDA is a neurotransmitter. It stands for NMDA is an abbreviation for N-methyl deaspartate. Morphine is an opioid analgesic, and ketamine is an anesthetic. Okay, so we'll be connecting morphine and ketamine to NMDA, and we'll also be specifying our search result. Okay, so we'll go on the build feature. We'll select a tool. We'll go to Path Explorer. Okay, we'll then go ahead and add morphine and ketamine to set A, and we'll go ahead and add NMDA to set B. Okay, and we're going from set A to set B here. Okay, we'll go ahead and apply those changes right here, right? But prior to doing that, we'll specify for publications between January 1980 to December 2024, okay? And we'll select specifically for relationship types that are activation, causation, correlation, or expression, right? So we specify for this. Then we'll go ahead and click apply right here. And this shows us all the interrelated, all the relationships that IP has found for us, right? In this case, we'll just select for all of them and we'll add it to our pathway. It looks something like this, right? And we can go ahead and move the different nodes around to make it look a little bit more visually appealing like this, right? So that's basically, like how you would go about, just showing you how it would look if you wanted to filter your results. Okay. Now, the different layout options that we have available for us. So there are several layout options that IPA provides for us, these being the organic, the circular, the radial, hierarchical, and subcellular. We'll be going over how we would go about changing the layout and how the different layout options look right now. Okay. So we'll just continue with our example from last time, right? So we'll just use this example and we'll see how the different layout options would look for this example. So morphine and ketamine connected to NMDA, okay? So if you wanted to see how to change the layout, we click on this button right here, which is right beside the expand button right here, right? So this button over here, this is how we change our layout. We click on that. It takes us to this right here. And we're basically able to select whichever layout we want. So that being organic, circular, radial, hierarchical, subcellular, right? We'll begin with organic. So how that would look. So an organic layout will look something like this. So we have NMDA receptor over here in the middle, right? Just sort of an array, so, sort of like this, right? If you wanted to arrange it in a circular layout, it will look something like this. So we have all the different molecules / nodes on the periphery, and we're able to visualize all the different relationships. Like, so all the different lines are able to be visualized with this layout design like this, okay? Next, the radial layout. So the radial layout, we have NMDA receptor on the center and all the different molecules / nodes on the periphery like this. We have the subcellular
Hi,
layout. Yes.
excuse me. Those molecule outside.
Where? Molecule outside. Let me just check the chat. Maybe you wrote something there. The name of the software is IPA. Okay. All right, we'll just get back onto it. Okay, so this is the radial layout. We also have subcellular, and basically subcellular, it basically shows you, it basically categorizes the pathway based off the subcellular location of the different molecules. So for example, if a molecule is located in the extracellular space, if it's located within the plasma, if it's located within the cytoplasm, or if it's located within the nucleus. So like, it might be a little bit difficult to see, right? But basically this over here is extracellular. Let me use the laser pointer. Okay, so this right here is the extracellular space, right? So all the molecules that are found within the extracellular space are over here. The plasma membrane, so all of them that are within the plasma membrane are located over here. All the ones that are found within the cytoplasm are located over here, and the ones that are located within the nucleus are found here. So IPA go ahead and lays it out, your pathway, in a way that, if you want to basically visualize where the different molecules are located, it basically formats the pathway in a way like this, so that you're able to visualize which subcellular space each of the different molecules are found. Okay. And also the hierarchical is another method by which you can sort your data. So this is what IPA generated for us, right? So I just decided to include this basically just to show you that oftentimes the way that the layout is arranged for an IPA isn't exactly visually appealing or practical, right? So you can see the different molecules / nodes are very far apart, and it's quite impractical, right? But I've just included just to show you that, oftentimes the layout options are not practical, right? And, at the end of the day, arranging it the way that you see fit is, what you should be doing, right? Again, it's about how you want to fit it into your research, or how you want to, basically assort the different nodes in your pathway, right? So this is how IPA produced the hierarchical layout, and it's quite impractical, as you can see. But oftentimes it's not like this, right? This is another pathway that's arranged in a hierarchical method, and this is obviously quite a bit more visually, like congruent. It doesn't look, quite a bit like this. The nodes aren't far apart or anything, right? So it really depends, how you want to go about doing it. You do have this tool available to you, right? That you can change the layout, but again, it's upon your decision. It's your call, essentially, how you want to lay out the information. Okay. And the shortest paths and the shortest path plus one as well. Okay, so basically the shortest path. So if you remember from Path Explorer, when we have our different pathways, our different relationships over here, right? You might have noticed that there is shortest paths on the right side over here, right? But if you click on shortest paths, it will also show us the shortest paths plus one. So basically the shortest paths, in a nutshell, it is basically the least number of nodes. They can go from set A and set B, or set B to set A, right? So it is the least number of nodes that are between them. So in our previous example, we did ketamine and morphine related to NMDA, right? So in the shortest paths, we found that there were two nodes in between, right? So the shortest paths basically just shows you however many relationships have the least number of intermediary nodes. So with ketamine and ketamine and morphine to NMDA, the least number of nodes that IPA could produce for us was 2, okay? But in shortest path plus 1, it's able to show us 3. So shortest path plus 1 is just one additional intermediary node to the shortest paths. The shortest path is just the least number of intermediary nodes. So in this case, ketamine, L-glutamic acid, NMDA receptor to NMDA, right? This is 2 intermediary nodes right here, right? So that is the shortest paths. And shortest paths plus 1 is going to be 3. So in this case, if we go from morphine to cyclic AMP to PKA, NMDA receptor to NMDA, right? This is 3 over here, right? So that's the basic difference, right? And essentially, right, if whichever you want to include, right, that option is available for you, because typically shortest paths have far fewer relationships, right? Shortest paths plus 1 typically have a lot more, right? So we'll just go ahead and show you an example of using the shortest paths and shortest paths plus 1, how that may differ in its appearance. So we'll use the example of morphine and ketamine connected to IL-1. Okay, we'll go on to build, right? We'll add morphine and ketamine over here. We'll select for morphine and ketamine, right? We'll add it to our set A. We'll select for IL-1. We'll add that to set B. Okay. Then we'll also use our specifications as well, right? So we'll specify this data for the nervous system specifically, and activation, causation, correlation, and expression relationship types. We'll go ahead and apply those changes. And we get this over here, like the shortest paths, there's only one interaction. So we'll go ahead and add that to our pathway. We get something that looks like this, right? Then we'll also check the shortest paths plus 1. Okay, so in this case, with the shortest paths, there's only one interaction over here, right? But if you go to the shortest paths plus 1, we see that there's an additional 10 relations, okay? So we'll see what relationships are added with the shortest paths plus 1. So we see that there's an additional 10. We can just go ahead and select for all of them, add it to our pathway. And I've already gone ahead and rearranged the pathway. So it's something like this. So we can see that with the shortest path plus 1, there is in fact an interaction with morphine and ketamine to IL-1. Prior to that, like shortest paths, it just showed us that morphine had a connection with IL-1, right? So that's something that you're able to do, right? If you see that there is no relationship prior, to using just with shortest paths, right? You're able to go on to shortest paths as 1 to see if there's actually any interactions that were not shown in shortest paths. So just keep that in mind. And finally, we'll be going over how to export your data. So this is especially important if you want to make a publication. You might want to export your data because it does include the Kaizen Kaizen logo at the bottom. So that oftentimes can be important. So to export your data, basically what you do is that you click on this button right here, right? This takes you to this page right here. You're able to specify for the format of the image that you want. So that being JPEG, PNG, etc., right? You're able to format the image as you like, right? Then you can click OK over here. Upon clicking OK, that will take you to this page right here. You're able to select the location that you want your file to go to. Over here, once you've selected the location, you can type in the name. In this case, I've named it morphine ketamine relations to IL-1, right? Then you can go ahead and save your data. And that's basically how you go ahead and export your data. So we'll take this time to answer any questions. If anyone has any questions regarding anything in this session.
Hi everyone.
Yep.
Yeah, I'm sorry. This is my first session, so I'm not quite too familiar with the IPA software. How can I get it? How can I get access to it? Is it free source or so?
Yeah. So basically, there is a free trial. So during the session, we will be utilizing the free trial from IPA. I have outlined, in fact, I'll just go over this again for you and like for anyone else that may be joining us today. I'll just show you how you can go ahead and apply for IPA. So I have included how to sign up on our Google Drive. I'll
Okay.
link Drive as well. Do you have the Google Drive link already?
Um, so I do have it.
You do have it, okay. I'll just go over it again, just so you have an understanding. If you have any questions as well on how to sign up, I'll just address that right now. So basically, in order to sign up, you can either follow this link over here, right? Or you can also type in on your browser IPA Kaizen, and it's going to be the first link basically that you click on, right? Upon clicking that link, it takes you to this page right here, right? And on this page, you click on request a trial right here. Upon clicking that, it takes you to this page right here. And basically you just have to fill this out, right? And just have to type in your first name, your last name, company, this being your school. You type in position. So in this case, it'd be student, undergraduate student, or high school student, right? You type in your institutional email. This is your school email, right? You type in your phone number. You type in your country, and then you can type in using IPA. You can just click on other, or you can click on biomarker identification. It doesn't really matter too much, right? It's just something that's also asked in IPA as well, right? So you can click on other or another, option, right? And then you can just go ahead and submit your what's it called? Your application for IPA. This is also on the Google Drive, by the way. So I can link the Google Drive right now, if you'd like, in case someone else doesn't have it. But all of the presentations, as well as how to sign up for IPA, how to use the Grow feature, the Path Explorer feature, that is all included into the Google Drive. So that's the link for the Google Drive right there.
So may I ask?
Yes, any other questions?
So I want to ask one question.
Yep.
Sir, the assignment which you gave in the last session,
Yes.
so sir, what is the deadline of that submission, and how can we make that assignment? In our phone, can we make that assignment?
So you would need a computer for the software. So there's no real deadline. It's just more so for your practice, right? It's just so you can get to use the software, right? And basically, again, like it's one thing explaining it, right? But if you actually utilize the software, right, the understanding that you get from it is different from explaining it. So if you weren't able to complete it, you can just complete it by the next session. That's fine as well. But I don't believe you can use your phone. You would need a computer in order to use the software.
Sir, is it compulsory to submit for all?
It would be beneficial, right, to submit, but it's not compulsory, right? But you know, if you're able to submit it, that would be preferred.
Okay, sir. Thank you.
All right, any other questions that anyone has?
Yeah, can you hear me?
Yeah.
Could you go over the shortest path plus one function once more?
Oh, sorry, could you repeat the question?
Could you go over the shortest path plus one function once more?
Yes, I can go over that, yeah. So the shortest paths plus one. So basically, like I mentioned before, it's basically shortest paths is the least number of nodes, specifically in the Path Explorer. So if you go back to Path Explorer, in fact, I'll just explain with that. Start with that, in fact. So yeah, so in Path Explorer right here, right? So when we select for what set A is, what set B is, right? We click apply, we get these different relationships over here, right? So we can see that there's shortest paths right here, right? But if you click on this, we'll also get shortest paths plus one. So the shortest paths is basically the least number of nodes that you can go from set set A to set B, or set B to set A, right? So buprenorphine over here to apoptosis, the least number of nodes in between, that is what shortest path is going to show you, right? Shortest path plus one will show you one additional node from shortest paths. So instead of one node, there's going to be two nodes. So that's the basic difference between the two. That go back to shortest paths right here. Yeah, that it'll show you just like the least number of nodes in between, and shortest path plus one will show you one additional node to the least number of nodes. That's basically how you can think of it. So in this case, right, like we have ketamine and morphine connected to NMDA. The least number of nodes that we can have in this case is two, right? So that's what it's going to show you all the different relationships that have two nodes in between. But shortest path plus one is going to show you three nodes in between. So that's the basic difference, right? And then like we went ahead and connected this using Path Explorer, right? We got just one over here, right? So we have two nodes, right? If you're connecting morphine and ketamine to IL-1. But if you check for shortest path plus one, we get 10, okay? So, and shortest path plus one, it shows us three nodes right here, whereas shortest paths was just two nodes in between. That's the basic difference. I hope that improved your understanding of it.
Yeah, it did. Thanks.
Okay, no problem. Any other questions? Okay, I guess we can go on to the quiz now. So I will be sharing the game. In fact, so this is the QR code for the game, for the Kahoot. You can go ahead and scan the QR code on your phones right now. Then I'll be loading up the game pin shortly. Okay, so upon scanning the QR code, you should be taken to a page that looks like this. So over here, you just have to enter in your game pin, which I will be showing up on our screen right now. All right, so the game pin is right here. I'll also copy the game link onto the chat. Sorry, give me one sec. Okay, so the game pin is on the chat as well. You can go directly to that. We'll give a few minutes for everyone to join. Okay, is anyone not able to join yet? We can leave, we can give a bit more time as well right now. Does anyone need more time to join? Okay. All right, we can begin in a little while. If anyone else doesn't need to join, we'll just begin right now. Okay. All right, is everyone ready? I can begin now.
Sir, can you please wait for at least for 1 minute?
Yeah, that's fine. Yeah, we can wait until, yeah, we can wait 2 minutes. Yeah, it's fine.
Sir, can you wait for 2 minutes?
Yeah, that's fine. Okay, can I begin, or do you still need more time?
Sir, please wait because my internet is very slow.
Oh, okay. Okay, I think around an additional minute, then I'll just begin then, because there are a couple of people leaving as well. So we'll begin in around a minute. Okay, so we're going to be starting now. All right.
What does the shortest path in Path Explorer shows us?
Okay.
Sir, please bring the passport.
Sorry.
Sir, where's the passport?
I'll check in the chat after this last question. Okay. Okay.
Sir, I asked about the passport.
Okay. Yes, you asked something, sorry. I'll check in the chat if you might have written in there. Oh, sorry, you were asking something before.
Nothing, sir. Your was not showing the passport. That's why I was not able to join the Kahoot.
Oh, okay. I because of network issues. I could maybe there will be a final quiz, by the way. So this isn't the final quiz. Um and the monetary prize is part of the final quiz. So it's not the end of it's not the end of anything. I could maybe share with you guys the questions as well if you wanted for your review. But I'm really sorry that any network issues you might have had weren't able to join the quiz. Okay, so I'll make a note of whoever won the whoever came first, second, third for today's quiz. And additionally, we do have a task for the next session. So the task is so I want you to use the Path Explorer function to connect calcium and potassium both. So calcium, potassium, they will be set A, connected to osteolysis, and select osteolysis specifically as a connective tissue disorder, right? And I want you to specify for publications between January 1980 to December 2023. And I want you to also specify a filter for activation, correlation, expression, transcription, and relationship types. So these 4 relationship types I want you to filter for. So specifically these ones. And also publications between January 1980 to December 2023. So I want you to do that, filter the results using these specifications. And additionally, I would also like you to then change the layout of the pathway to circular. So the layout options that we discussed, right? Change the layout to circular. And additionally, I would also like you to use the Path Explorer function to connect ethanol and morphine to pentraxin 3. And I want you to specify it for macrophages, activation, causation, correlation, expression, and transcription relationship types. So these what's called these 5 relationship types, and specifically in macrophages as well, right? And I want you to show it in the shortest paths first, then add shortest path plus 1 to the pathway. Okay. So initially how it looks in shortest paths, then add shortest paths plus 1 and compare how the pathway looks with shortest paths first, then shortest paths plus 1. Additionally, I've also included the task in the Google Drive link. And if you're able to submit the task into the Google Drive, I have created a what's called a Dropbox right here. So the task is in the Google Dropbox, in the Google Drive rather. So the task that is to be completed is right here. And if you're able to submit your work into the folder, that would be it's more so for your benefit as well. So you're able to utilize the software. And if you have any questions for the next session, we can address those questions as well related to using the software. But that is the task for the next session. It's also found in our Google Drive link. I'll just include it into the chat for the Zoom call as well. So if anyone wants to look at it right now. Any questions related to the task or any other questions that anyone has? So that is the end of our session then, if no one has any other questions. Thank you all for attending today's session. And I hope to see everyone for the next session, which is going to be on August 24th, next Saturday at 11 a.m. Eastern time. And hope to see you all then for our next session.